Hello, good morning. It is a great pleasure to present our researcher today, and I would like to thank the organizing committee for giving me this wonderful opportunity to present here today. So my name is Zvi Hayouka, and I'm from the Institute of Food Science and Nutrition from the Hebrew University of Jerusalem from Israel. And today I will tell you about our research on the development and exploring the novel approach for developing novel antimicrobial peptide. First, I would like to share with you some insight about the Hebrew University of Jerusalem. So this is in Israel, very tiny country in the Middle East. And for the Hebrew University of Jerusalem, we have four campuses. Three of them are located in Jerusalem, and one is located in the farther side, in the central, near Tel Aviv. And this is our Agriculture, Food, and Environment faculty. In Jerusalem, we have the En-Karen Medical Center, Hadassah, that all the teaching for the medic studies and humanities in the Mount Scopus, and the natural science. This is the main campus that's located in Givat Ram in Jerusalem. And you can see here we have variable views in Israel, and I will invite all of you to come and visit us here in Israel. The antibiotic resistance crisis is a threat in the global health and also for food security and all developing countries and all over the world, actually. The misuse of antibiotics in humans and animals is accelerating this process, and there is growing number of infections are becoming harder to treat or unable to treat. And this is a very frightening era. In addition to all the COVID-19 crisis, it's even become worse because not all of the virus infection is known as a virus, and then it might be treated as an infection of bacteria. And then there is another cause for misuse of antibiotic, which accelerate the antibiotic resistance. My lab focus is mainly to develop novel antimicrobial peptide that will have a very low, or if any, rate of resistance selection. So I will tell you today in my talk about our two main projects that are running in my lab. The first one is the random antimicrobial peptide mixture approach and how we developed this approach and why we developed it. And also another very different approach, how we use Quorum sensing system to inhibit the cell-cell communication pathway in bacteria in order to prevent the bacteria from being pathogenic to a non-pathogenic state. There are many examples of antimicrobial peptides that are being secreted all on our body and actually in all the organisms that are existing on Earth. This is part of their innate immune system in order to fight the first fight and to prevent infections, any microbe infection. And this race started in the early 80s, where several researchers found the gene that are being translated to a very short protein, a very short peptide actually, that are able to eradicate or to prevent the growth of bacteria. And from that moment, there are many examples of antimicrobial peptides that were discovered and characterized in the literature. And you can see here that several examples of antimicrobial peptide, and in the last 30 years, many researchers worked and used several approaches as peptidomimetic in order to enhance the stability and also the activity of this peptide. But I really like this slide because you can see here how broad the antimicrobial peptides in terms of their structures. You can see we can have alpha helical structure, beta sheet, or a random coil peptide from several organisms as frog, human, worm. And what is very interesting for me when I see this picture is to realize why nature bothered to design this amazing structure if all of them have the same antimicrobial property. All of them interact, most of them interact with the bacterial membrane and cause a damage on the membrane and cause a leakage of the component inside to the outside, and in that way they kill the bacteria. So what we did, we wanted to see, okay, so the difference, the structure are totally different, and also the sequences are totally diverse. So what is the common features? Like, how can we explain the antimicrobial properties of this compound? And what we observe that all of them have net positive charge, at least of +2, and this may explain the first mode of action. This is an electrostatic interaction with the negatively charged bacterial membrane with the antimicrobial peptide. So without having this positively charged, there is no interaction. So this is the first important step in order to enhance or to induce the cell growth or the cell death. The other feature is that we observed that all the antimicrobial peptides that were discovered so far contain at least 50% of hydrophobic amino acid. And that may explain the second step of the antimicrobial property. The first step is the interaction, but then all the hydrophilic part interact with the lipid chain of the bacterial membrane in order to disturb it and to cause and induce the bacterial cell death. So keeping that in mind, that there are two common features, the positively charged and the hydrophobic moiety, we asked ourselves, okay, so do we really need a defined sequence? In most cases, we have antimicrobial peptide, natural antimicrobial peptides that in aqueous solution, they don't have any structure. But upon binding to the membrane, they fold mainly to amphiphilic helix, which one side is hydrophobic and the other one is the cationic. And then you can imagine that the peptide the helical peptide can interact with the bacterial membrane with the phase of the cationic and then make a flip-flop inside the bacterial membrane in order to induce the disruption and to induce the bacterial cells death. So in order to answer this question, if we really need defined sequence, we used a combinatorial chemistry approach, and we developed a new type of antimicrobial peptide. We term them random peptide mixtures or random antimicrobial, as I will show you soon. And the main idea is we use the regular or the conventional solid phase peptide synthesis approach, but instead of incorporating one amino acid on the solid support in order to have a one sequence in our peptide, we decided to use two amino acids, a mixture of two amino acids, that one of them is the hydrophobic and the second one is the cationic, because this is the only features that were common for all the antimicrobial properties. And then we can generate many different types of peptides on the same bead. And you can see here, after one step, we will have two options. After two steps, we will have 2 in the power of 2. So we will have 4 optional sequences on our mixture. And if we will continue to 3 steps, we will have 2 in the power of 3. That means that we will have 8 optional sequences, or on the same bead or in the other solid support. But at the end, to make a very long story short, at the end, we synthesize usually 20-mer peptide. This is the optimal chain length that we observed great antimicrobial property. At the end of the synthesis, we cleave the peptide with an acidic condition, and then we generate 2 in the power of 20, that means 1 million optional sequence in our pool. And this approach, we use this compound. Of course, we characterize them by using Malditov mass spectrometry and amino acid analysis to make sure that the ratio between the hydrophobic and the cationic amino acid is well preserved. And in this approach, we can very easily change the composition of the peptide, the cationic and the hydrophobic amino acid. We can easily control the chain length because we can perform how many steps of coupling on the solid support. And what is very interesting, we explored the stereochemistry of our peptide and how it affects the antimicrobial properties. And as you can see here, we can summarize here several peptide, random peptide. You can see here in the x-axis several mixture, leucine and lysine, phenylalanine and lysine, isoleucine and lysine, or with an arginine. All of them is 1-to-1 molar ratio between the hydrophobic and the cationic amino acid. This is the optimal ratios that we observe. If you will increase it to more cationic, you will decrease the antimicrobial properties. And if we will increase the hydrophobic, it will aggregate and it will be very insoluble. But for 50-50, 1-to-1 molar ratio, we observed great antimicrobial properties on several compositions. In the y-axis, you can see this is the minimal inhibition concentration, and these values mean that how much compound we need from to add to our sample in order to prevent the growth of the bacteria. So as these values are lower, that means that the compound is much more potent. And you can see here, very surprisingly, that the leucine lysine and phenylalanine lysine able to prevent the growth of gram-negative E. coli or gram-positive B. subtilis, or even antibiotic-resistant strains that were selected from humans. This is a methicillin-resistant Staph aureus or vancomycin-resistant E. Physium. And we can see that the activity is very broad towards gram-negative and also towards gram-positive. And most of the natural antimicrobial peptides that were discovered so far from nature are able to eradicate or gram-negative or gram-positive. So we were really surprised to see that we have great compound towards gram-negative, positive, and also for antibiotic-resistant strain. When you design an antimicrobial peptide, it's very important to design a very selective compound because we don't want to cause any damage to the host and just to eradicate the bacterial cell membrane. And what we observed here, we did an hemolysis study in order to see how our compound is able to select between prokaryotic membrane or eukaryotic membrane. And what we can see here is that unfortunately, in a very low concentration, of our compound, we can see that we have great hemolysis. So that means that this compound, the phenylalanine lysine, in 1-to-1 molar ratio as a 20-mer peptide, is not so selective. And of course, this was very unfortunate. But as an organic chemist, I'm really interested and admire all the stereochemistries that we have in nature. And what we did, we explored how the stereochemistry of our peptide are able to change the pattern or the selectivity of our compound. So far I showed you our results about the L-homochiral, the natural amino acid, but we synthesized D-homochiral, we use D-phenylalanine and D-lysine, or heterochiral mixture or random stereoisomers. And I teach organic chemistry class, 500 students each year, and the way that they are really remember the stereochemistry is using this joke that Samuel L. Jackson and Samuel D. Jackson. So this is very important. For the antimicrobial property, we could not see any effect, and this is very reasonable for us because there is no chiral specificity. This is not protein-protein interaction, that the chirality of your compound is very important. But here, this is just electrostatic interaction and hydrophobic interaction in order to induce the bacterial cell death. But what we were really amazed to see is the hemolytic activity profiles that we observed for these stereoisomers. So as you can see here in red and in black, this is the homochiral L and D, which we showed previously that were really hemolytic, and you can see here. But when we altered one of the amino acid of the mixture to D-amino acid, or D-phenylalanine, or D-lysine, we totally abrogated this hemolytic activity. That means that these compounds are really selective, and we did not affect the antimicrobial property. So that means that the heterochiral mixture are much more selective, but preserve the great antimicrobial property. And our main hypothesis was that the homochiral mixture may self-assemble to a structure that is more toxic to the eukaryotic membrane, and by using non-natural amino acid, we prevent this structure to form, and by that, we prevent the hemolytic activity. And in order to support this hypothesis, what we did, we took the red mixture, the homochiral L and the homochiral D, and before doing the experiment, we mixed them together in 1-to-1 molar ratio to get the same concentration, and we wanted to see the effect on the hemolytic activity. And amazingly, we observed, this is the green line, you can see that we observed a great reduction in the hemolytic activity profile just by mixing the two homochiral mixture together. And our assumption that because we mix them together, we prevent the formation of the toxic pathogen. The last stereoisomer is the racemic mixture. When we use racemic phenylalanine and racemic mixture of lysine in order to synthesize this peptide. So what we can conclude for now is that we have a very selective compound and very efficient in terms of antimicrobial property. But for me, as an organic chemist, I was amazed to see that one chiral center, the D-amino acid, totally changed the hemolytic activity profile. And what we wanted to know is to shed light on the mode of action and to dig more inside in order to visualize or to be able to learn about the mode of action of this peptide. So what we did, we initiate a collaboration with the National Physics Laboratory in the lab of Max Rianda, and we used atomic force microscopy where we mobilized on the mica a lipid bilayer that is supposed to mimic a bacterial membrane. So that means that this is more negatively charged, so we add more DLPC, more than the DLP gene, ratio of 3-to-1, in order to have a negatively charged lipid bilayer on the surface. And then we added our peptides, and we incubated them, and then visualized the topography of the surface. And what we were really amazed to see is the ability of the homochiral mixture to self-assemble to a pore, a very uniform pore with 200 nanometer diameter, and it was amazing for us, and we put it on our refrigerator for a very long time, this picture, because we were really proud of it. And we synthesized this peptide also with isotopically labeled N15 phenylalanine in order to visualize and to do a complementary assay where we use the secondary ion mass spectrometry in order to visualize the location of the peptide. And what is amazing here to see that they are self-assembled, all the peptides are concentrated inside the pores in order to cause the leakage and the damage to the bacterial membrane. But what was more interesting for us is no matter what we did, we could not observe this structure when we used our heterochiral mixture, the D-lysine and the N15 isotopically labeled phenylalanine. No matter which condition, how many time, the concentration, we could not visualize any self-assembled of this structure. In the secondary ion mass spectrometry, you can see that the peptides are all over the place. And the conclusion from this part is that when we altered the stereocenter, we totally changed the mode of action, the antimicrobial mode of action of this peptide. In the area of the antimicrobial peptide, there are mainly a pore-forming mode of action by causing a pore in the membrane and causing a leakage. But there is another interesting way of killing the bacteria, which called a carpet model. This is Yehiel Shai from the Weizmann Institute from Israel, has explored and suggested this interesting mode of action in the early 90s. And the hypothesis is that the peptide interact with the membrane, all over the membrane, till it causes a collapse of all the membrane, and then by that, causing the leakage of the bacterial membrane. And what we did, we wanted to explore more deeply of the kinetics of the differences between the homochiral and the heterochiral mixture. So before we started, we assumed that the self-assembly is a process that is much more faster, because it's more uniform and it's more thermodynamically favors. And the carpet model will take more time for the bacteria to do their mode of action. And amazingly, you can see here, we did a killing assay when we took an E. coli bacterial cells, 10 in the 7th. We incubate them in PBS with 4 micrograms per ml of our compound, or homochiral or heterochiral. And you can see in 4 micrograms per ml, after 10 minutes of incubation, we could not see a huge effect. But in 20 micrograms per ml, we can see that most of the homochiral mixture totally kill the bacteria. There is no survival. And heterochiral, it's less active. So that means that for the carpet model, it takes more time in order to kill the bacterial cells and to cause the damage. We use fluorescently labeled, and we visualized the peptide, and what we observe that they are totally penetrate into the bacterial cells, and they might have more other activities inside the bacterial cells, and that's what we are exploring right now in my lab. Another great challenge in the area of antimicrobial peptide or tackling pathogenic bacteria is the ability to inhibit or to eradicate mature biofilm. Biofilm structure of bacteria start when several cells, planktonic cells, adhere on a surface, start replicating, start expressing extracting several carbohydrate, extracellular matrix with proteins, with carbohydrate, and it becomes much more challenging to treat this structure. And there are several reasons, but first, of course, this is the physical membrane that you need to penetrate in order to kill the bacteria from the inside of the biofilm. But the other thing that is really interesting is that on the biofilm, the bacteria become entering a growth arrest. So there is no DNA synthesis, there is no RNA synthesis, there is no cell wall synthesis. And when they enter in the growth arrest, all the conventional antibiotics actually do not act on this bacteria because there is no synthesis of RNA or DNA. So all the mode of action of the conventional antibiotic is totally abolished, or there is no need. So this is a great challenge also in the food industry, but also in the biomedical devices. So what we showed here is that when we took mature biofilm, and when we took mature biofilm, that means that we grow the bacteria on a plate for 48 hours, and then we observed a thick biofilm on the bottom of the plate, and we added our compound for 24 hours. And what we measure is the biofilm biomass by crystal violet staining, and you can see that at a very low concentration, 25 microgram per ml, we could hardly see any biofilm biomass, so there is a great reduction in the biomass of the bacteria on the surfaces upon the treatment. In order to visualize it, we used scanning electron microscopy, and you can see here the control. This is the biofilm without any treatment, and of course, after the treatment of the homochiral or the heterochiral mixture, we could hardly see cells, and most of the structure are degraded, and these bacteria cannot overcome the treatment. That means that even if we wait a week after, they won't be able to form a new biofilm. Recently, we started to work on Listeria monocytogenes, a very, very challenging bacteria. And I think the main challenging part is that these bacteria can survive and multiply even in low temperature, in the refrigerator, in 4 degrees, and it becomes much more challenging to treat this kind of bacteria. And Listeria is also able to form a biofilm that also makes it even more challenging to treat. And what we showed that if we will use a conventional antibiotic on the biofilm structure that we obtained from Listeria, you can see by using ampicillin, we cannot see any effect on the biofilm biomass, but when we added, this is a 200 microgram per ml of the peptide, and this is from the reverse side of the concentration, you can see that we got a great in a degradation of the mature biofilm, and we support it also using by scanning electron microscopy. As you can see here, the control compared to the treated. This is with a leucine-lysine random peptide mixture, but we observe the same pattern also with phenylalanine and mixture. So I will conclude for now that the random peptide mixture our approach has very interesting activity. So you can see that they are really active and broad activity, and they have also anti-biofilm activity. But I think they have two main advantages. The first one, that they are easy and cheap to synthesize. And this is very important, because if we want to use it in biomedical devices or in pharma or in clinics or in the food industry, this is very important, and it might be a great issue all the cost. But the second advantage is because we are dealing with a cocktail of peptide, our assumption that it will be very challenging for the bacteria to develop resistance toward this bacteria. So for this compound. And in order to answer this hypothesis, we are running many experimental evolution studies in my lab when we expose the bacteria for our peptide in cycles. So every day we are changing the media and adding a fresh media with fresh amount of peptide. And what we measure the sensitivity or the activity of the compound, the MIC value, the minimal inhibition concentration, and we would like to see if there is any change in these values along the time. And what we observed very easily is that bacteria become very easily resistant to the antibiotic. This is a rifampicin, but for the random peptide mixture, we could not see any effect or any dramatic change, and this is what we are doing right now, and I hope that at the end we will show it soon, how it's important, and we need to do it more time and much more, many more cycles. For the first time now, we are starting to work on a mice model to see how it affects in animal model, and the first model that we're using is to treat mastitis in lactating mice instead of lactating cow, and to see the effect of the random peptide mixture, but more importantly is to see the safety of our compound in animal study, because so far we showed that on cell line we could not see any cytotoxicity up to 500 microgram per ml of our peptide, no matter if it's homochiral or heterochiral. So of course we know that mastitis is a common disease in breastfeeding women, and also of course in dairy animal. It's caused by E. coli and many other type of bacteria as mycoplasma, and there is a constant need to develop a new way to treat mastitis. And the most important part is that it's very difficult to treat with an antibiotic, because then the milk of the cow will contain high amount of antibiotic, and then we accelerating the exposure of human that consuming the milk for an antibiotic with no need, and then we are enhancing the resistance occurrence. So the main idea is to develop a non-antibiotic treatment to treat mastitis, and this is what we are doing right now. And the first step was to first see that there is no effect on the mammary epithelial cells. So we did many cytotoxicity, and we labeled the peptide, and we could see that there is no effect on the mammary gland epithelial cell, EPH4 cell line. The next step, we used IVs. We labeled our peptide with indocyanine green, that has a very interesting 700 nanometer excitation and emission. And then we can see very easily using all body imager on the mice without sacrificing them. And then we can have more pharmacokinetics parameters and to visualize where is our peptide after we administrated them on the mammary through the teat canal. And you can see here that they are really concentrated on the mammary gland. This is just the bacteria. This is just the peptide without any infection. And we could see them also in other organ. We assume that the mice is leaking, and then they eat the peptide. But what we did then is we visualize and quantify the safety of this treatment. And what you can see here is that the mammary gland is very healthy, even in high concentration. When we administrated even 1 milligram per ml of the peptide. And when we did the infection, that's what we did actually last week, we observed great reduction in the bacteria burden in this mammary gland. So we observed great antimicrobial activity in the mammary gland, but this is a very preliminary result, and I hope that next time I will share with you some more statistical and some more results on this regard. And actually my lab is mainly also, I'm from the Food Science Institute, so we are also having a lot of efforts to improve food safety and also to extend food shelf life. So what we are doing, we're working on several directions in order to prevent food loss and to extend food shelf life. So we started from the basic. We have several projects to purify water from bacteria. We have a project on crop protection that we use our random peptide mixture on the greenhouse in order to show that there is a great reduction in the disease progression in plants. We use our peptide as a food preservative because when we use homochiral mixture with L-amino acid, with natural amino acid, we assume that they will totally degrade in our stomach, in the gastric fluid, but then will not affect the consumer. And we are really interested in developing bioactive packaging. But in order to design bioactive packaging or to design bioactive surfaces, no matter for food or for biomedical devices, we need to decorate surfaces using our random peptide mixture. And the main hypothesis of the project was if we will be able to decorate a surface and to preserve the antimicrobial activity of the peptide, and this is the main challenging part, we will have an approach when the bacteria will interact with the surface, will become damaged. As you can see here, the red bacteria, this is a damaged bacteria, and then it will be killed. And in that way, we can prevent the contamination of the surface. So what we did, we changed the chemistry of the synthesis, and we synthesized the peptide, and then we can decorate any surfaces, if it's polystyrene, if it's a glass, if it's a silicone wafer, and many, many other examples. And when we immobilize the leucine lysine, phenylalanine lysine, or tryptophan lysine, we observe that the leucine lysine have more activity than the other examples. So we decided to focus on the leucine lysine random peptide mixture. And here you can see when we took the surface that was decorated with the leucine lysine peptide, you can see the relation or the dependence on the microbial load on the activity of the surface. So up to 10 in the 6th of bacterial cells, we could observe great antimicrobial activity, but if we increase the microbial load to 10 in the 7th or 10 in the 8th, we can not see any activity. And of course, it depends on the surface of the surface, the size of the surface, and the loading of your the antimicrobial peptide. We showed that we have a great activity towards several pathogenic bacteria, as methicillin-resistant Staph aureus, which is gram-positive, Pseudomonas aeruginosa, which is gram-negative, and E. coli. We totally degrade eradicate all the bacterial cells that were exposed to this surface. In order to shed more light on the mode of action, we took the beads that we decorate with our peptide, and you can see here, this is the beads, the polystyrene beads without our peptide, and you can hardly see a bacteria on them. But when we decorated our beads with the random peptide mixture, we can see that the bacteria adhere or absorb on the bead by electrostatic interaction. And if we will incubate it for a while, we can see the great damage that is caused to the bacterial cells. Here, they're treated with the random peptide, immobilized random peptide. You can see most of the cells are totally broken or damaged. Here, we use the live dead staining, when the green one is the live bacteria, and the red, this is the damage, the membrane damaged bacteria that will be killed at the end. And you can see here, this is the bead without the peptide. You can see that most of the bacteria still green, but when we will incubate them with the immobilized random peptide mixture beads, you can see that most of the bacteria that on the top of the surface are being damaged, and at the end, they will be killed. This is along the time. You can see that it's more red. That means that more bacteria are being damaged. What we continued to do is we decided that we can use this method, these beads, to purify a column, microbial contamination of water. And we were amazed to see that a very basic column that we developed in the lab, and we put in the water a PBS with an E. coli microbial load of 10 to the 5, to the 5th, or to the 4th, and measure the microbial load on the inlet of this column, and you can see that we can get a great reduction, and all the bacterial cells are being killed or degraded, and the water are totally pure at the end without any bacterial cells. Actually, we just initiated a new startup company based on this technology, Peptide. It came in order to purify and to protect our source of life. This is in collaboration with Strauss, a huge food company here in Israel. And the main idea is to be able to perform passive or non-thermal pasteurization of water and juice and many other examples of liquid. So far, I showed you our project on the random peptide mixture, but we have another interesting project on the Quorum sensing system, and I see that I think my time is running. I will just go briefly on that, and I will show you how we are able to design a selective peptide with one specific sequence and not random in order to prevent a cell-cell communication pathway in bacteria. And this is in collaboration with Didier Lacrosse from the National Institute of Agricultural Research in France, from INRA, and Leila Slamati is a research assistant. So if we will prevent cell-cell communication or the Wi-Fi, this is not my kids, but most of the kids will react like that. And I will go briefly on that, but there are many bacteria that will express a virulence factor just when they will sense a community of bacteria on the environment, and then they will start together to express and secrete a toxin to the environment. Because if one bacteria will secrete, we will not have any effect on the host. So the idea is that they become a community, and then they will have much more effect on the host and the environment. And the first example is the Staph aureus quorum sensing system, which contain this cyclic peptide, thiolactone peptide. And we worked on B. cereus. This is a foodborne pathogen, and it has a 7-mer peptide that is being used as a sensor for detecting the microbial load of the bacteria in the environment. And what we did in this project, I will go very briefly because I see that my time is running. I will skip, and I will just show you at the end what we observed here. So these bacteria, when they become community together, they can secrete a hemolysin. This is a toxin that can hemolyze red blood cells. And what you can see here, that when we added our compound to the supernatant, to the growth media of the peptide, we can see that there is a great reduction in the hemolysis expression and secretion. Because you can see here, this is totally dependent on the concentration of the peptide that the bacteria was exposed to. But this is a peptide that we could not see any effect. So you can see that the hemolytic activity is totally without any effect, as when we did not add any peptide to the environment. And then we did it also on Bacillus cereus, the ATCC strain, and we could observe a much dramatic effect. And by that, you can see here that this is without our peptide, and this is with the increasing concentration of the peptide that we designed. You can see that there is a great reduction in the hemolytic activity. So that means that there is no expression of virulence factor, and that means that we totally block the cell-cell communication pathway in this bacteria. And this is our first paper when I asked my student to add this non-Wi-Fi in order to show that there is no cell-cell communication. And we took a bacteria that is a pathogenic, and we forced it to become non-pathogenic. But the bacteria could not detect it because they still grow. So there is no evolutionary stress to develop resistance toward this compound. And this is, I think, the most important part in this area of the quorum quenching. And we did another study when we studied more deeply the mode of action and the contribution of each amino acid of the peptide on the activity on this cell-cell communication pathway inhibition. So if you would like to read more, you can read our paper, and I'm sorry because I'm running out of time. So we showed that this is an antivirulence peptide. They can totally block the quorum sensing, and they can prevent all the expression of virulence factor. So first, at the end, this is the thanks. And first, I need to thank all my group members that are really great, and they are doing a great job to all of them. This is Abhishek, this is the quorum quenching project, this is hers. And all of this part are working on the random peptide mixture. I would like to thank, of course, the funding. And I would like to thank you for your attention. And it was a great pleasure. And if you need more questions or if you would like to collaborate, I will be more than happy. So you can send me an email and we can be in touch. So thank you very much. I wish I could come and see you and meet you and discuss with you. But unfortunately, in this time, we could not do that. So I will really appreciate if you have time to contact me. Thank you very much.
Thank you very much. All right, there is a lot of question in here, but since the Dr. Hayouka is in Israel. He can answer, but I read a few questions in here. I think it's very interesting. Can these random peptide mixture be designed to work against envelovirus? And then another comment is amazing talk, very impressive. Bacterial resistance is a very important topic, and then also the agricultural application is very important and interesting. Indeed, in Singapore and other countries, there right now emerging technology after nanotechnology, biotechnology, food science and technology. This is new field. It's different from food nutrition. This is a topic for the whole food security.